So, hello. So we're going to start the session this afternoon. It's very exciting about metabolic diseases. So we have very good people talking for actually 5 different talks. And we're going to start with Dr. Nicola Longo from the University of Utah. He's a professor of pediatrics as well, chief of medical genetics. He's going to talk about carnitine and fatty acid oxidation.
Thank you, Dr. Megawa, for the introduction. Can you hear me? Thank you. So it's a real pleasure and honor to be here. So the first time I attended the SERGG meeting was in 1986 in Navarre Beach, and it was just a few years ago. So in my career, I was at Emory from 1985 until 2001, and then I moved to the University of Utah. And my interest in carnitine and fatty acid oxidation started while I was still at Emory University. And what I have to, this is my disclosure, and I've been working with a company in Italy that makes carnitine, and that is what you have to keep in mind during my presentation, but I will not discuss any unlabeled use of carnitine. So what I want to do today is to describe carnitine and what is its role in fatty acid oxidation, how disorders of fatty acid oxidation are diagnosed, the treatment of fatty acid oxidation disorder, but mostly of primary carnitine deficiency, and the role of newborn screening in the detection of fatty acid oxidation defect. And actually, there is a talk just after mine that will discuss the diagnosis of some of these disorders. So what is carnitine? Carnitine is a chemical that looks like an amino acid, except that it has 3 methyl groups directly on the nitrogen that was initially isolated from meat. Meat is carne in Latin, and that is why it is called carnitine. And people here that are of Hispanic origin know that carne is meat. Carne is also meat in Italian. So why there is so much carnitine inside meat? Because the muscle burns a lot of fat, and carnitine is necessary for fatty acid oxidation. Now, how do we get carnitine? So obviously, we can get carnitine in the diet, and the vast majority of carnitine, about 75%, is either from meat or dairy products. If you think to children, to babies actually get most of their carnitine from the diet because they also have a limited capacity of synthesizing them, because the body is capable of synthesizing carnitine. The carnitine is lost in urine, and one of the, what we see in acute renal failure, we see that many patients have very high level of carnitine. This is what we see in acute renal failure. By contrast, patients who have in chronic renal failure and are on dialysis can have very low carnitine level. But going back to the synthesis, babies have very limited capacity of synthesizing carnitine. And in fact, what we have seen in recent years, there are formulas that are devoid of carnitine, and some of these babies have tested positive with the newborn screening simply because they were receiving carnitine formulas without any carnitine. How is carnitine synthesized? Carnitine is synthesized in many organisms, and this is a schematic of the synthesis of carnitine, keeping in mind that some of the latest steps only happen in certain organs. What is important of this schematic is that when we started having patients with very low carnitine level, we were thinking that in theory, a defect in the synthesis of carnitine could lead to carnitine deficiency. Now we know that it is not the case because we have identified, not myself in particular, but defects in the synthesis of carnitine have been identified, and specifically a defect in the enzyme here, which is called 3-hydroxylysine hydroxylase, has been 3-methyl lysine hydroxylase has been identified. And it is important because the gene for this enzyme is on the X chromosome. And what they found exactly, they found that a deletion of one or more than one exon in this gene is associated with non-syndromic autism. And it has been proposed that the deletion of this gene is a risk factor for autism in the general population. Obviously, since this gene is on the X chromosome, the deletion of this gene would explain in part why autism is more frequent in males than it is in females. What also they found that it is still not clear what is the mechanism by which this deletion can cause autism or predispose to it. What has been found is that in children, in older children with this condition, the level of carnitine in blood are usually normal to low normal. And also there is an elevation of other chemical prior to the synthesis of this compound in the metabolic pathway. So it is not clear whether the predisposition to autism would be given to the low carnitine level or more likely to the accumulation of the chemical before the synthesis that accumulates in this condition. What has been found also that at least in one family, the administration of carnitine supplement has improved the autistic symptom in children affected with this condition. So this is the very first defect in carnitine biosynthesis that has been identified, and more recently, a second defect has been identified, and in this case, there was a full gene deletion in one of the last steps in carnitine biosynthesis. In this case, also, there is the accumulation of a precursor prior to the metabolic block and mild carnitine deficiency. However, the phenotype in this case was difficult to ascertain because it was part of a contiguous gene syndrome, so we do not know whether the symptoms observed in the child were due to deletion of this gene or deletion of one of the genes next to it. Now, what is the main function of carnitine? We discussed that carnitine is important for the fatty acid oxidation, and this slide shows a schematic of fatty acid oxidation. So usually, fatty acids do not mix with water. As a result, they need to bind to proteins to be transported in the general circulation. Once they reach the tissue where they need to be utilized, what they do, they enter the cell through the action of specific fatty acid transporter. There is a complex system of protein-protein interaction that makes it in a way that the fatty acid never get exposed to water, but just enter the cell through the fatty acid transporter. Once it is inside the cell, the fatty acid, which can be extremely toxic, is immediately conjugated to coenzyme A and sequestered within the cell. Once the fatty acid is bound to coenzyme A, it forms a very large molecule that cannot diffuse anywhere. At this point, the fatty acid can reach the organelles that are devoted to fatty acid metabolism, the peroxisome, the microsome, or mitochondria. In physiological conditions, the vast majority of fatty acid oxidation occurs within mitochondria that also are responsible for energy production. The other two organelles also metabolize fatty acid, but they do not produce any energy. So if we want ATP, fatty acid has to go to mitochondria. Now, as I told you previously, the fatty acid bound to coenzyme A doesn't move around very well. So the only way by which the fatty acid can enter mitochondria is through conjugation with carnitine. Carnitine enters the cell through the action of a specific carnitine transporter, which is called OCTN2, organic cation transporter number 2, the novel type. And once carnitine is inside the cell, will be conjugated to the fatty acid, to the acyl group, by the action of CPT1, which is carnitine palmitoyl transferase 1, which is located in the inner aspect of the outer mitochondrial membrane. As a result, the fatty acid will attach to the hydroxyl group of carnitine, forming an acyl carnitine, and then the acyl carnitine can enter the cell through the action of the carnitine acid-carnitine translocator. Once the acyl carnitine is inside the cell, the enzyme called carnitine palmitoyltransferase 2 can do the reverse reaction and attach the acyl group, the fatty acid, to coenzyme A. And at this point, the acyl-CoA is ready for fatty acid oxidation. At the same time, the release of the acyl group will free carnitine again, and the free carnitine will be able to return to the cytoplasm to the action of the same carnitine-acid-carnitine translocator to initiate a new cycle. Once the fatty acid, the acyl-CoA, is inside mitochondria, it will undergo a series of reactions that are responsible for fatty acid oxidation, starting for the enzyme responsible for the oxidation of very long-chain fatty acid, and then through the action of the three functional protein, will lead to the shortening of two carbon atoms from the fatty acid with the formation of acetyl-CoA. Two molecules of acetyl-CoA can combine together to form ketone bodies, and the shortened fatty acid can undergo another cycle to do further shortening and further generation of two carbon atom units that will undergo oxidation within the Krebs cycle. Now, when do we get symptoms from fatty acid oxidation? So we get symptoms when fatty acids are necessary to produce energy. And normally this happens during fasting. When we eat, the excess energy is stored in the adipose tissue. And when we fast, these fatty acids are released from the adipose tissue and reach different organs, among which the liver, the heart, and the skeletal muscle. In the liver, fatty acids are not fully oxidized, but they will generate ketone bodies. The ketone bodies are as good as glucose in producing energy, and they can reach the heart, the skeletal muscle, and also the brain. The fatty acid cannot reach the brain directly, but through the liver they can generate energy usable by the brain through the formation of ketone bodies. Now, what happens when fatty acid oxidation is defective? When fatty acid oxidation is defective, fatty acid will still be released from the adipose tissue and accumulate in organs that will not be able to metabolize them, especially in the liver. The liver will show severe accumulation of fatty acid, or what we call hepatic steatosis, which is micro and macrovesicular. At the same point, the formation of ketone bodies will be impaired, and for this reason, all of the tissue will utilize all of the glucose that it is available, and we have hypoketotic hypoglycemia, which can lead to loss of consciousness, leading to coma and subsequently death. In addition, the fatty acid can accumulate inside the heart, and they can have a direct toxic effect with cardiac arrhythmia or cardiomyopathy. In the muscle, they can produce muscle breakdown, and with myoglobinuria, in some cases just mild hypotonia or a myopathy. Now, what do we see if a patient dies during an acute attack of fatty acid oxidation? We see a couple of things. First of all, if we look at the liver, we see the accumulation of fat, which are these white vesicles that would stain very well with Sudan red or any other fat staining if one had used that one. The second thing that can be seen is the depletion of glycogen store. This one in the big slide is PET staining of tissue from the liver, of a patient with primary carnitine deficiency. And this is the normal liver of a person who is doing perfectly fine. And the blue staining is the glycogen. If you starve a normal person, there will still be some glycogen, and the lower image shows just a normal person after fasting. In a patient with fatty acid oxidation, all of the glycogen is completely gone, and there is no residual glycogen. And this happens both in the liver and the heart. So, and this is responsible for several of the pathophysiology of fatty acid oxidation disorder. So there are two main mechanisms. One is the lack of energy, and the second mechanism is the toxic effect of accumulated free fatty acid. Now, the disease that I've been studying for a long time is called primary carnitine deficiency. And the frequency is about 1 in 142,000 in the United States, as ascertained by newborn screening. There are some other regions, such as the Faroe Islands, where the frequency is 1 in 200, 1 in 300 individuals, which is obviously much higher. It is due to a deficiency of the carnitine transporter. And what happened in this condition is that the lack of the carnitine transporter results in carnitine wasting in the urine and carnitine deficiency in every tissue. As a result of that, there will be the inability for carnitine to bring fatty acid inside mitochondria and secondary impairment of fatty acid oxidation. There are different types of presentations that we will review in a minute. And the other thing, how do we diagnose it? We diagnose it because there are very low levels of plasma carnitine in this patient, usually less than 5 micromolar. And we confirm the diagnosis either by functional studies or by DNA testing. The therapy consists in the administration of carnitine that needs to be continued for life. We monitor free and total carnitine in this patient, and the prognosis is excellent as long as treatment is continued for life. If people can stop treatment, they can have sudden death and die. Now, let me go back a moment to the clinical presentation. And the clinical presentation is important because this child, I saw this child while I was at Emory University. And I entered the field because my colleague did not believe that this child had primary carnitine deficiency. So this is a child who presented at 8 months of age with a history of frequent infection and vomiting. And then at one point he became lethargic and he was admitted to the hospital. He was found to have a big liver and had non-ketotic hypoglycemia and elevated ammonia. So as a result of the elevated ammonia, he was placed on a low-protein diet because he was believed to have a urea cycle defect and sent to see genetics. So when we examine this child, what we notice is that the carnitine level were extremely low. So the normal is for total carnitine 30 to 70, and the level was 1. Both parents had mildly reduced carnitine levels, suggesting carrier status. And obviously, we started giving carnitine to this child that, to my knowledge, is still doing very well. Now, the presentation of this child is hepatic encephalopathy, where we have a non-ketotic or hypoketotic hypoglycemia, and it is observed very frequently in children very early in life, up to about 2, 3, 4 years, sometimes even 6 years of age, but very early in life. In older children, what we can see is cardiomyopathy. And this is a child that was referred to us from an outside hospital because the child had this history of cardiomyopathy and the resistant to any type of therapy. The child was listed for a heart transplant, and during the evaluation, he was found to have very low carnitine level. This child was found to be homozygous for a mutation in the carnitine transporter gene, started on carnitine, and the cardiomyopathy went away. So obviously, this is the second type of presentation that we see in children with primary carnitine deficiency. So hepatic encephalopathy and cardiomyopathy. Both presentations can occur within the same family, among siblings. So there is no real genotype-phenotype correlation in this aspect. Meaning that what we see, we just see that one can have either presentation independently from the mutation. And this is what we have been thinking for many years, until we started doing newborn screening for primary carnitine deficiency. And then we started seeing other aspects of primary carnitine deficiency. And this is a patient, actually, that was referred to us from Greenwood Genetics Center, from Dr. Irene DeBiase and Dr. Tim Wood. And this is a young lady who had a history of syncope for many, many years. Nobody could understand why. And she had long QT syndrome. And then at one point, she became pregnant. And the syncope were due to cardiac arrest, so she had a placement of an automatic defibrillator. And during pregnancy, the level of carnitine dropped. She had several attacks of cardiac arrest during pregnancy. After the baby was born, the baby was found to have very low carnitine level. which led to the diagnosis of primary carnitine deficiency in the mother, and the mother was started on primary carnitine deficiency, and to my knowledge, she hasn't had any other attack, and the long QT syndrome resolved with carnitine supplementation. So this is the other type of presentation of primary carnitine deficiency, and we were not aware of this one until we started doing newborn screening. So people can present at any age with sudden cardiac arrest, rhythm disturbances, less likely cardiomyopathy. And this can present really at any age. Other symptoms that have been found in asymptomatic adults are easy fatigability, decreased stamina, exercise intolerance, fasting intolerance, and especially palpitations are very frequent in patients with primary carnitine deficiency. But many patients, many adults with primary carnitine deficiency are absolutely asymptomatic, but they are at risk of sudden death. In other words, they can die without any symptom, and if we do an autopsy in them, we do not find any accumulation of fatty acid. So in other words, the autopsy would not give away the diagnosis. Let me go back a moment to diagnostic confirmation. The best system to confirm the diagnosis remains a functional assay. And this just shows carnitine transport in fibroblasts of normal control and patients with primary carnitine deficiency. And you can see that the carnitine transport in these fibroblasts is reduced to 20% or less of normal. And that was the only system of making the diagnosis until the gene was identified about 15 years ago. And the gene is on chromosome 5, and it is composed of 10 exons, and then encodes a protein with 12 transmembrane spanning domain. And this transmembrane spanning domain, what they do, they form a pore through which carnitine can enter inside the cell. And over the years, we have identified many mutations in this gene, so there is not a single mutation, but there are just many, many different mutations that are heterogeneous and differ from family to family. And what these mutations, many of them are missense mutations, and I believe that this slide only shows the missense mutation that we have identified, and they impair activity of the transporter. Now, we have many people here who are part of CleanGen, and obviously you want some type of demonstration once you find a mutation to connect that with the disease. How do we make this demonstration? So what we do, we express the carnitine transporter in Chinese hamster ovary cell. We express obviously the normal one that here appears as a green molecule, and if this transporter is effective, we'll be able to transport carnitine inside the cell. If it is not effective, it will not be able to do so. And this shows just a sample of mutations that we have expressed in the carnitine transporter to determine whether they are pathogenic or not. These are all missense mutations. And you can see that some of them retain nearly completely normal carnitine transport activity. Obviously, those are not mutations, but that they are just polymorphism. This remains one of the best ways to determine if a mutation is pathogenic or not. And this mutation just gives you an update of mutations that we have identified, with the yellow one being the new mutations that have never been reported. Now, many times people do not have the advantage of having the expression study to determine if a mutation is pathogenic or not. And they recur to programs like Polyphen or SIFT to determine whether mutations are pathogenic or not. And their prediction is about 70 to 75% correct. So in 25 to 30% of the cases, the mutation cannot be predicted correctly whether this is pathogenic or non-pathogenic. So one has to keep that in mind to determine the pathogenicity of the mutation. And the second thing that we found is that obviously if we have a functional assay, we know that the patient is affected. And we found that in about 16% of the allele, we could not identify any specific mutation. Obviously, we are not sequencing the whole gene. We only sequence the coding region of this gene. So we design primer for each exon we amplify the exon and the flanking region, and we sequence that region. So there are mutations in the intronic region or in the promoter region that cause primary carnitine deficiency, and they account for about 15% of the pathogenic allele that cause this condition. So as I told you before, there is no correlation between genotype and phenotype in children. So in other words, you can have cardiomyopathy or hepatic encephalopathy, and there is no correlation between the type of mutation that you have and the type of presentation. It depends much more on environmental stressor, because obviously, being a fatty acid oxidation disorder, it is usually triggered by conditions that promote fasting. In general, infection are the most common condition that cause fasting in children, because children, when they are ill, they do not want to eat, and they need to utilize fatty acid to produce energy. Obviously, if a child has an infection early in life, will be prone to hepatic encephalopathy. If a child less than 6, 7 years of age without an infection can develop cardiomyopathy. Now, even though there is no correlation with the early presentation, we found that the people identified by newborn screening, such as the mother of our patient with primary carnitine deficiency, have significantly higher residual carnitine transport activity as compared to people presenting symptomatically. So retaining residual carnitine transport activity seems a little protective against the childhood presentation of primary carnitine deficiency. So how do we know that this mother will have symptoms? So the way that we know is because there is, in the Faroe Islands, which is an archipelago between Scotland and Iceland, there is a very high frequency of this condition. About 1 in 200, 1 in 300 people are affected. And what happened a few years ago, about 15 years ago, they noticed that there were a lot of young people, age 21 to 35, who died suddenly. So you have, you have about 50,000 people living in the Faroe Islands, and then you have a dozen of them that all of a sudden die for no reason. And they found that they had primary carnitine deficiency. So this led to a nationwide screening program in which people of all ages were tested for carnitine level. So they identified a lot of people with primary carnitine deficiency, but the prevalence of these people decreased as people age. What it means is that you had a high prevalence early in life, and then the prevalence decreased because these people died. So as a result of the screening, a lot of people have been placed on carnitine, and these episodes of sudden death have disappeared. Most of those people were completely asymptomatic before that. Now, what is the therapy? The therapy is carnitine administration that we use not only in primary carnitine deficiency, but also in secondary carnitine deficiency. And usually what we do, we give carnitine, and then we monitor plasma-free and total carnitine. This is usually well tolerated. Some people develop gastrointestinal symptoms, and you can see that in the illustrative, in the prescribing information, seizures are also described, probably because carnitine is given to a lot of patients with a variety of diseases, including diseases where seizures are relatively frequent. By itself, I've never seen it causing seizure. The most common side effect is that it can cause a fishy odor. What it means that people can develop this bad smell, and obviously this can prevent compliance because people do not like to smell like rotten fish. Why do people develop this type of symptom? So first of all, when you take carnitine, the carnitine that is present in food is absorbed relatively efficiently. About 50 to 75% is absorbed. If you take with supplement, only 15% is absorbed. Why? Because we are giving a large dose all at once, and a lot of carnitine remains in the gut. What happens to that carnitine? That carnitine can be transformed in trimethylamine, and trimethylamine can be reabsorbed by the intestine and transformed into trimethylamine oxide. trimethylamine is responsible for the smell of rotten fish. And obviously what we need to do, we need to try to remove that one because obviously people do not like to have it. How do we decrease the side effects of carnitine, especially in our patients? There is no single strategy. But the most important strategy is to divide the carnitine administration in 3 to 6 times per day. The second thing that I found useful is that the dose that we recommended in the past, 100, 200, 300 milligrams per kilogram per day, is way too much. Usually I stop at 100 to 150 milligrams per kilogram per day. I just increase the number of daily administration because that it is way more effective than increasing the dose. The second thing, I tell all patients to take yogurt or cultural or something that has active yeast because those ones reestablish the proper microbiome in the gut and decrease the degradation of carnitine to produce trimethylamine. In resistant cases, I give short courses of antibiotic, of metronidazole, that kills anaerobic bacteria and can help restoring the normal flora. And in other cases, if that doesn't work either, then we even can stop carnitine. One has to keep in mind that even in patients with primary carnitine deficiency, what is important is the chronic assumption of carnitine. If one stops it for a few days, nothing happens. And that is part of the reason why some of these patients become non-compliant, because you stop it, you notice absolutely nothing whatsoever. You not notice nothing for a week, sometimes nothing for a month, and sometimes children or adults have sudden death after a month to 2 months or 3 months without being on carnitine. That is why it is important to reinforce the need for taking carnitine in this patient. And the last strategy is the use of strong deodorant. They can actually cover the smell. And the best ones are the ones that are used by hunters that usually can buy in hunting store because they really cover your smell very effectively. Now, the OCTN2 carnitine transporter is encoded by this gene, which is called SLC22A5. And what you find is that if you look in the exact browser, you find a lot of variants. Obviously, in the exact browser, you only have carriers, but that can give you an idea of what is the carrier rate of this for mutation in this gene in the general population. Since we have expressed so many missense mutations, obviously we assume that the missense mutations that we have demonstrated to be pathogenic are pathogenic. And the second thing, we assume the stop codon and splice variant are pathogenic as well. Now, the calculated frequency of the disease, if we substitute those numbers, is 1 in 80,000. And instead, the rate of identification of patients with primary carnitine deficiency in the United States is 1 in 142,000. Why do we have this discrepancy? The reason is that when we measure carnitine in the baby, the carnitine level depends on the level that are present in the mom. And the closer we are at birth, the more they will reflect maternal level than they reflect the baby's level. And on the left here, you have the level in patients that were subsequently confirmed to have primary carnitine deficiency. You can see that at birth, the level of carnitine were above the cutoff, which is the red line. And then they drop slowly below the cutoff, allowing their identification. By contrast, if you have a mother with primary carnitine deficiency, the levels are very low immediately at birth. And then, if you give carnitine, this is in the baby, okay? And then if you give carnitine to the baby, the level rise very quickly and very briskly, simply because the baby does not have primary carnitine deficiency, but in most cases is only a carrier of primary carnitine deficiency. So that is why babies can be missed by newborn screening. Now, the main reason we believe is that when children are tested too early, they can be missed. We have tried all of the possible algorithms, including doing where you sum up many of the acyl carnitine species, but no matter the way that you do, some babies who have normal carnitine level at birth are normal whichever algorithm you use to try to identify them. So in other words, it is a biologically normal sample. So you need to wait for time for carnitine to decrease. Obviously, there are states that do two newborn screening, one at birth and one later in life, and obviously it is likely that the second screening is more effective in identifying them, even though we don't have still data to confirm this type of assumption. What I know is that, for example, in the Faroe Islands, where we know the specific mutation causing the disease, about 1 out of 3 children with primary carnitine deficiency can be missed by the newborn screening. And the newborn screening there is done between 48 and 72 hours of life. So the newborn screening between 2 and 3 days of life can miss about 1/3 of patients with primary carnitine deficiency. And this is becoming evident both from the study in the Faroe Islands and from our study on the SERGG browser and the frequency in the United States. Obviously, DNA testing would be ideal, but would lead to the screen. In the United States, it wouldn't be feasible simply because there are way too many mutations that need to be identified to confirm or exclude primary carnitine deficiency. In summary, we have seen that carnitine is essential for fatty acid oxidation, and its deficiency can present at any age when energy from fat is needed. So usually during fasting, infections, fever, and patients are perfectly normal between episodes. The OCTN2 carnitine transporter deficiency causes a spectrum of diseases, from hepatic encephalopathy with hypoketotic hypoglycemia to cardiomyopathy to sudden death. And the sudden death, again, can occur at any age. Newborn screening can miss babies with primary carnitine deficiency, in part due to the timing of the screening. DNA testing identifies about 85% of causative mutation, with the rest being in non-exonic region. Carnitine therapy, continuous for life, can present clinical symptoms in primary carnitine deficiency, and we are just at the beginning on identify other possible role of carnitine in the physiology of human, and what has been found is that defect in carnitine biosynthesis have been identified as a risk factor for non-dysmorphic autism, and it results in low normal carnitine level with accumulation of precursor before the metabolic block. And again, they might represent a risk factor for non-dysmorphic autism. I want to thank all of the people in my laboratory and in the clinical lab and in the clinic who have contributed to have allowed us to collect all data on this patient, and also and especially all of the patients and the family with primary carnitine deficiency that have participated in our study. And here it is shown just some views of Salt Lake City, the University of Utah Hospital, and ARUP laboratory of the University of Utah. Thank you very much.
Nicola, that was a great lecture. Thank you. You know, in terms of screening for autism, the people who do these screenings are always asking us, what should we send when patients present? Autism is really common, supposedly in the United States, 1 in 60 or whatever, very different than the numbers you would expect from this. How do you respond to them in terms of, should we be doing acylcarnitine profiles, even if they were done in newborn screening, on all children with autism? Do you promote that or not, and why?
No, I do not. So in autistic children, actually, I think that this is just one of the risk factors. So Art Baudet has been the main promoter of this association, it's not me. So I think that what I find important is, number one, we still do not know the mechanism. So it might be just the chemical that accumulate in this patient. And obviously when you give carnitine, you stop the synthesis of carnitine, which leading to a reduction in the chemical accumulated before the metabolic block. Now, obviously, one of the biggest problems I find is that some parents of children with autism would go to any length into finding any possible therapy for their patient. And obviously, this contributes to them being prey of people that can sell them any type of supplement that it is believed to be useful. I think that's what we need to do. We need a little bit more study to determine exactly how this predisposition works. And one of the theories of Dr. Baudet is that if you have an association of this defect in synthesis with decreased supply of carnitine early in life, you may actually increase the risk of autism. Because obviously, there are vulnerable periods of the brain, especially early in life, where the brain can be wired to be autistic or not. Obviously, that it is the most critical period. And one troubling trend that I'm seeing is that more and more there are these organic formula which are supplemented with a variety of things, but not with carnitine. And I do not know how you get supplemental riboflavin and other things which is organic in this formula, but that's okay. But they do not contain carnitine, and we have case after case of children that come in with an abnormal newborn screening because they have normal level of carnitine at birth. They receive 0 carnitine. Again, their synthesis is very minimal in the first few months of life. So they drop, the carnitine level drop, and then they come in and they say, what's going on? And we find that they have this formula. Obviously, if you associate an environmental factor like that one to the lack of capacity of synthesis, then you would be able to, to increase the susceptibility of children to autism. That's what I think it is happening. Either that one or children have a second mutation, maybe in the carnitine transporter or in another gene. Obviously, this is what we think. We don't have any demonstration. Does carnitine hurt in children? Probably adding carnitine to cereal, like we've done with folic acid, wouldn't hurt either. But babies do not eat cereal anyhow, so the baby's baby. So, but at the same time, we need to understand more the possible connection and how one leads to the other. So that's what I'm saying. So.
I also really enjoyed listening to that, Nicola. It really helped pull a lot of different things I knew about carnitine into one nice bundle. The question I had, I know there's some reports about potential carriers or patients that were found to be deficient in carnitine, but only found one mutation, and they were described as carriers in the literature. You're saying that they have a mutation that couldn't be found? And then the question is, do you know where in the gene that is? You think promoter? You think it's a del dupe? What do you think is going on?
I didn't hear the whole question. Let me get close to you so I can hear, okay? Sorry. So the question is about the carrier?
You said, I remember hearing that some carriers might have do you think they're carriers, or do you just think we haven't found that second mutation? And then where might that second mutation be?
Yes, there is one paper, not by me, where they have said that carrier can be at risk of certain diseases. I haven't found that, to be honest with you. And we did a study a few years ago because we suspected that cardiomyopathy could be more frequent in adults carrier of primary carnitine deficiency. We didn't find an association. We actually found that there was absolutely no association between cardiomyopathy and carrier status. Now, said that, there are patients that where the second mutation is not identifiable. Going back to the Faroe Islands, in the Faroe Islands there are 3 prevalent mutations. The most common is called N32F, which is the same one that your patient had, the one in Greenwood. That it is by far the most frequent and the most deleterious. The second one is a splicing mutation, which is a change 52 nucleotide upstream from the beginning of an exon. And the third one is called the risk haplotype. Risk haplotype because they know all of the polymorphins that are in cis with the mutation, but the mutation has not been found. They haven't done genome sequencing yet, and quite honestly, I would be willing to do it for them because I would like to figure out which one is the region that it is important for regulating the expression of this gene. Now, these other mutations are not as bad as the N32S. What I mean is that if you are homozygous for the N32S, you have about 2 to 3%, 4% normal transport activity. If you are compound heterozygous between the N32S and one of the other two, you have about 15% transport activity. You still get very low carnitine level with 15% transport activity. And in fact now, we call that, when we do functional studies, anybody with less than 20% normal activity is called affected simply because we know that they cannot maintain a level of free carnitine above 10 without supplement. So, but obviously we cannot exclude the possibility of a second allele unless functional studies are done. So obviously I do not have the patients that were reported as carrier, and they cannot tell you one way or another. The only other thing that I can say about the autism is that we have identified autistic children in family of children with primary carnitine deficiency. And these children were affected. So in other words, some children were identified because they presented symptomatically with either cardiomyopathy or with hepatic encephalopathy. And some of their siblings were still asymptomatic, but they were delayed in development. And many of them improved dramatically after being placed on carnitine. Obviously, here we have a very clear-cut explanation, but again, we still do not know, however, the connection between carnitine and autism, if there is one.
Further questions? So we're going to move on to the next talk.